Okay, we're going to start in a second. I just need to get my pointer going. And let's go. Basically, in chapter number 24, we're going to look at how kind of organisms defend themselves. Again, what we're looking at is a vaccine is a harmless derivative variant of disease-causing microbe. Vaccinations against HIV, the virus that causes AIDS, have not been successful. There's a number of reasons for that, and again, a little bit of it is changed. In the past, what we found is that we can get harmless versions of a pathogen and use it, and it would work. And in this case, it didn't. And so we'll get more into that detail. What we call immunity is basically the body's defense against disease. This is protection against pathogens, and pathogens are viruses and other microorganisms. And so again, we've done a number of things to protect ourselves from very large predators. These kind of small predators that we can't see, we would call pathogens. We have a number of lines of defense against, and so we have our external barriers and behaviors. We have what are called our innate defenses, which are present at birth regardless of whether we've seen the disease before, and what we call an adaptive defense, which is activated only after exposure to specific invaders. And so in our body's defense, these innate defenses and these adaptive defenses, what we see in innate defenses, we have things like our skin, our secretions, mucous membranes, phagocytic cells, neutral killer cells, etc. These are all things that work constantly all the time to protect us from pathogens. The second thing that we have, an adaptive defenses, we have the production of antibodies. We have certain cell types, B cells and T cells, that kind of help protect us from pathogens that we've seen or things that we've been exposed to in our environment. And we'll be able to talk about these in a little bit. Again, in looking at our external innate defenses, our skin, the secretions, spit, mucus, urine, the mucous membranes, these are all things that kind of act like barriers or packaging that prevent microorganisms from getting inside of us. Yet, again, we can't protect from everything, and every once in a while we'll get a thorn or something poked into us. And so what we have is a set of what we call phagocytic cells, and these are cells that will engulf, phagocytize, basically to consume or to eat. And so here's a little pathogen, and the cells wrap themselves around this pathogen and destroy the pathogen. And these types of cells are what we call nutricellular cells. They produce defensive proteins, and they are part of what we call our inflammatory response. And so an inflammatory response is just basically any response that will occur for these organisms. Okay. In the adaptive response, basically we see the production of antibodies, and this is the molecule that we want. So this is an antibody. An antibody is a protein that's made in response to a particular antigen, and we'll get a chance to talk about an antigen a little bit later. The lymphocytes are cells that actually use the antibodies as kind of marker cells in order to bind to various things, and T cells have a version of this as well. So again, the physical barriers of skin includes all the things that we talked about before. These are the external packaging that prevent microorganisms from getting inside of us. Again, a good internal, that is, once things get past our skin and inside of us, when things go inside of our body, get through our GI tract, what has to have happen is that the body needs to be able to detect things that are foreign to it, that are not normal to it. And it needs to distinguish non-self from self, and that's a big thing. The second line of defense that we see, so the first line of defense in our castle is that external barrier, and then once we're on the inside, we have a number of things that will protect us from this. These things include white blood cells and other defensive proteins. And so, again, how white blood cells work is they phagocytize cells, Neutral killer cells destroy infected body cells and cancer cells. Interferons protect the body against viral infection. And the complement cells will actually kill invading microorganisms. So there's a number of different things. And we talked about that. So what's going on is here, this is basically kind of a show of how interferon works. And what's going on with interferon is when a cell gets infected with a virus, the cell produces a signal that tells other cells to be a warning. There's a virus around. Make antiviral proteins and slow down. Next thing that we need to look at is this kind of non-specific defense. And so whenever your body gets assaulted by microorganisms, and again, here's a splinter coming in. This splinter has various bacteria on it. The bacteria signals. So all bacteria have the same kind of proteins, kind of these general pathogen proteins, things like the peptidoglycan, etc. And the body can sense these, and the macrophage can sense these. And so what happens, the macrophages exit the circulating blood cells and go and engulf the area. Damaged cells release chemicals that reduce blood flow to the damaged area, and an area is going to be warm and red. And so when you get vaccinated, a lot of times your shoulder hurts due to this kind of non-specific response. Anti-inflammatory drugs like aspirin and ibuprofen may short-term release the pain, but it also reduces the swelling and fever, which sometimes may be a good thing. The second thing I want to talk about is we really didn't talk about this during the circulatory system, and we should have, is we have another system that's kind of similar to lymphatic system, actually to our circulatory system, is what we call a lymphatic system. And so a lot of times you think of, I liken this, and I like this analogy, I liken this to like the sewer system. You pay your water bill. You don't pay your sewer bill, you pay your water bill. And we always think about water going into a house, but you never think about sewage going out of a house. You need to have that sewage coming out of the house. And the lymphatic system is like a sewer system. And so it's a branching network of vesicles that actually return liquid basically to the circulatory system. We have lymph nodes, we have a number of things. And so lymphatic systems, lymphatic vessels carry what we call this lymph, which is very similar to the interstitial fluid that surrounds the cells. And here's an individual, and this is showing basically the lymph vessels, and these are the lymph nodes. It's one of the reasons why the doctor will touch your inner throat and kind of your armpits and again the apex. And so what all these things are doing is taking fluids that are leaking out into your tissues and bringing those fluids back up to the vena cava. Okay. The lymphatic system is responsible for returning fluid back to the circulatory system. And so how our circulatory system works is that blood flows into the capillaries, the liquid flows out and becomes interstitial fluid, and some of that liquid will come back in, but not all of it, again, because of the pressure differences. So that extra fluid would build up in our legs and our arms and other appendages, but except that it's able to be picked up by the lymphatic system. The lymphatic system also helps fight infections, and I'll describe how that happens in a little bit. The adaptive defense is basically our third line of defense. And so you have a physical barrier, you have these non-specific defenses, and the adaptive defense is the very specific defense. And what this does is it recognizes and it responds to specific invading pathogens. This is responsible ability of two cells. We have the B cells, which mature in the bone marrow, called B, and the T cells, which mature in the thymus, which is a gland in the chest. They eventually make their way to our lymph nodes and other lymphatic organisms where they can, here's this bone marrow cells, the differentiation. So here's the T cells and the B cells. And what they do is they kind of hang out in our lymphatic system. And because fluids are moving very slow in our lymphatic system, they can monitor what cells and what proteins are going around. Let's take a break and talk a little bit about antigens. Antibodies are what bind to antigens. Antigens are what causes the body to produce antibodies. These are molecules on the surface of a virus's and foreign cells and other molecules, and they specifically elicit an immune response from the lymphocytes. And so this is the critical thing. An antigen is what does this. So how do these cells recognize that? Is basically the B and the T cells develop these antigen receptors on their surface, and each cell recognizes a single specific antigen. Okay, and then the great diversity of B cells and T cells produce enough different antigen receptors to bind about every single possible molecule that's out there. So when a particular B cell binds to a particular antigen, it gives rise to short-lived cells which secrete a receptor-like molecule that's called an antibody. And this is the antibody right here. The critical thing about the antibody is it's bivalent. And so it can polymerize because it's adding one thing to another thing. And then once it polymerizes a molecule, it pulls it out of solution. So with antibodies, so you need to know what antigens are. Now you need to know what antibodies. You saw a picture of an antibody before. These are Y-shaped molecules. They have binding sites with a tremendous variety, and they enable the immune system to react to just about any kind of antigen that we have. They combine with an antigen to form what we call the antigen-antibody complex. And so a big thing about this is what we call clonal selection. So you need to know what this is. This is clonal selection, which generates B cells and T cells that are appropriate to the invading antigen. And this is the mechanism that underlies the immune system's specificity and memory of antigens. So again, it's one of the things that I want you to know. So what's going on in clonal selection is that here, in this case here, we have 3 different B cells that are producing different antibodies. And what happens is when the antigen comes in, and this antigen is complementary to the antibody, right? What it does is it causes this particular cell to move more of itself. And so we don't really have 3 cells here. We have 3 million different cells. And so what this clonal selection does is pick that one cell that binds to an antigen and causes it to replicate more of itself. And what's going to happen is you get what are called effector cells that produce lots and lots of that antibody, and you get the memory cells. And again, these cells are going to live for hopefully the rest of your life, and they're going to protect you. If the body, the body may never ever get exposed to or need this particular set of B cells, but it may need this one all the time, and it's clonal selection that allows that one to be produced. And so here's a little bit more of kind of what's going on. And so let's say you get exposed to a particular antigen or you get vaccinated at day 0. What's going to happen is we're not actually going to see antibody production for at least 7 days. You don't see maximum antibody for roughly about 2 weeks after your exposure. Now, if you got a really bad disease, in 2 weeks you may be dead. In hours you may be dead. You can't produce this. But if you can survive long enough, you produce antibodies, and once you get over the disease, given a period of time, what happens is these antibodies kind of go away. When you get exposed a second time, rather than days, you basically have hours to get a response. And in days, you're going to get a very strong response. And so that's one of the reasons why a lot of diseases you get it once and you don't get it again. What's going on with vaccination is that you're tricking your immune system to think that it's seeing these disease-causing microorganisms and to responding to it beforehand. In the United States, the vaccines that we have has almost eliminated polio, mumps, and the smallpox. Smallpox has been gotten rid of. Now, can we have too much immunity? Yes, we can. Just like you can have too little, you can have too much. And so we see the interplay of immune response. Allergies are when we're sensitive to harmless antigens in the environment. So somebody is overstimulated by harmless antigens. Allergens are antigens that cause allergies. The symptoms of an allergy result from a two-stage sequence reaction. And so here's the pollen that somebody gets. You get your B cells that make antibodies. The antibodies attach to the mast cells. When the allergen binds to the mast cells, they degranulate and release histamine, which causes the swelling and the fluid and other things. And so what's going on a lot of times with an allergy, you're going into anaphylactic shock. And so what happens, the blood vessels become leaky. You leak interstitial fluid into the tissues. Your blood levels go down. And so this is an example of the EpiPen that again, groups use. Our immune system normally reacts only to foreign, or what we call non-self substances. It can attack its own tissue, but these are relatively rare cases. Examples of this include lupus, insulin-dependent diabetes, muscular sclerosis, and rheumatoid arthritis. Here's an example of rheumatoid arthritis. Immunodeficiency diseases result when one or more of the complements in the immune system are lacking. You're lacking the stage 1, stage 2, and stage 3. This leaves people susceptible to many different infections. An example of this would be SIDS, severe combined immunodeficiency, Hodgkin's types of cancers, and then from radiation and drug therapies that cause this. And again, the big one, AIDS, that everyone seems to know about. Okay, it's been about 15 minutes. I'm almost done. And so this chart here is looking at the innate defenses that we have. This is looking at the different lymphocytes. An example, close-up of a B cell and close-up of T cell. And this is basically looking at the antibodies that are produced by the B cells. And this is looking at clonal section, which you should be aware of. Effector B cells, I'm not going to get into that much. And then this graph here is definitely what do you want to know. This is showing you how